Hello, everyone, and welcome to our online lecture series for genetics. Today, we're following up our discussion of pedigrees last time and incorporation of some diseases to take a look at some Punnett square math. Because pretty soon here, Punnett squares are going to be more difficult to draw out than the math. In fact, by the end of today's lecture, we need to make sure that we fully understand the concept of meiosis and how independent assortment, after all, title of today's lecture is Assortment, how assortment of chromosomes at the metaphase plate has a huge impact on the overall gametes that can be produced. Because after all, we go from these situations where we have chromosomes being transferred and lining up at the metaphase plate to them being transferred in a Punnett square. Like in this Punnett square here, we had our autosomal dominant trait, capital R, lowercase r with lowercase r. We were able to see the probability of transmission of the uppercase R. It was 1/2 of the possible gametes here. That's supposed to be a 2. Whereas the other parent was always going to be contributing a little r with a frequency of 1. So you had a chance of getting a big R of 1/2, a little r of 1/2 from this parent, and you were always given this allele. This meant that you had a 1/2 chance of producing progeny that carried the big R allele. And indeed, in this pedigree, we saw that probability bear out. Now today we're going to be discussing expected probabilities at multiple loci, expected probabilities across multiple children, and how many children you might have to have in order to make sure that you produce progeny with specific gametes. So, let's eat this pie one slice at a time. The first topic that we're going to cover discusses product probabilities. Now, product probabilities we're going to first look at with 2 loci, because we need to put this visually into something that y'all will already recognize. Hopefully, you recognize this dihybrid cross from a previous biology lecture, looking at some of Mendel's peas in terms of color and the fact that they could be heterozygous at 2 loci. We're going to be focusing specifically on this F1 here, where we've got an individual that's heterozygous at both of our loci of interest. But we're going to change the nomenclature here, just so that way it's easier to read. We will have the 2 loci being capital R with a lowercase r, and capital G with a lowercase g. Now, just like we did our probabilities up here, where you had a 1/2 chance of selecting each, we know that with this given genotype, you've got a 1/2 chance of being a capital R and a 1/2 chance of being a lowercase r. Now, from there, our 2 possible options are pairing this uppercase R with a 1/2 chance for an uppercase G and a 1/2 chance for a lowercase g. The same is true for our lowercase r. This results in the 4 possible gametes of uppercase R, uppercase G, with a frequency of 1/4, 1/4 which you get by multiplying 1/2 times 1/2 to give you 1/4. Uppercase R with a lowercase g, again 1/4. Lowercase r, uppercase g, lowercase r, lowercase g. Now all of these frequencies should be very, very familiar to you because they're what we have when we look at our sides of our Punnett squares. So hopefully a good recap for you. You're definitely going to need to know this without any questioning in order to take on our next task, that being trihybrid crosses. So, in our trihybrid cross, we'll expand with another locus. Capital R, lowercase r, capital G, lowercase g, capital A, lowercase a. And let's perform a cross here, where we've got a trihybrid crossing against a trihybrid. Now, I don't really want to draw a three-dimensional Punnett square for this, right? Many times as a biologist, you will get these situations where you don't want to draw a Punnett square out, but it will benefit you to draw separate Punnett squares out for each of the possible combinations. So what do I mean by that? Well, let's say that you are interested in generating an individual that had a genotype of all capital letters. What is the number of crosses that you'd need to perform? Or rather here, what is the expected frequency of an individual who is all capital letters at all three loci? Well, we can do kind of a subsample of this to illustrate the process. Since we know that there is a rule of multiplication in between each of the frequencies of these loci, we can take the Punnett square for our R locus, and draw that out. Let's go ahead and fill this in. We can take this and identify how many have the genotype that we care about. Well, it's 1/4. Well, if 1/4 of the progeny at that locus are going to be of the genotype that we care about, and we want the exact same genotype under similar situations at our G and our A loci, we can take 1/4 times 1/4 times 1/4 and get our result of 1 out of 64, or 1/64th ratio in terms of the amount of progeny that we'd expect if we had, say, 1,000,000 progeny created by this cross. And while that sounds kind of ridiculous, keep in mind, there are corn breeding programs out there. Are there more than 1,000,000 corn plants on planet Earth? You betcha. So these, even though they seem kind of hyperbolic, these are statistics that you may have to work with in terms of your future as a biologist. So this hopefully gives you a good idea of what the product rule is. Now you could do the exact same thing here, given a different situation. Let's say that you were interested in the number of individuals that were heterozygous, right? So in terms of heterozygosity, we see that the frequency of heterozygotes in a trihybrid cross would be 1/2, and again, another 1/2, and another 1/2 at our three loci, giving you the result of 1/8. Now 1/8 is much, much larger as a number than 1/64th. And that's because you have twice as much probability of two times as many ways of producing these heterozygotes as you do with your homozygotes, all caps. From here, we're going to combine our product rule with the sum rule. When we're thinking about the sum rule, we'll take a little aside here and think about rolling a dice. When you're rolling a dice, let's say that you want to identify the probability of rolling a 4. You can write that out as the probability of one roll to get a 4 is equal to 1 out of 6 of those sides, right? There's 6 different faces to a 6-sided dice. I should say, maybe scoot this over and clarify. There are many more types of dice than just the 6-sided dice. So in rolling a 6-sided dice, you have a 1/6 chance of rolling a 4 exactly. Now, what's the probability of rolling 2 4s exactly? Well, when I roll one dice, does the result shown on the first dice have any impact on the result shown on the second? The answer here should be no, right? These dice roll independently. Because they are independent events, we use the product rule. This means that you want the 1/6 chance on the first dice times the 1/6 chance on the second dice, giving you a 1/36 chance overall. Now, things change when we want to indicate the probability of rolling 2 4s, or let's say that 4s will suffice, or 2 5s will suffice. Now, rolling in a 4 or a 5 can occur in 2 different ways on each dice. This means that you need to take the probability of rolling 2 4s, which we got as 1/36, and add it to our probability of rolling 2 5s. Because you can't roll both a 4 and a 5 on a dice in a single roll. Those independent events of rolling a 4 or a 5, or those non-independent events, of rolling a 4 or a 5, result in you adding things together. This, of course, gives you the outcome of 1/18. A much higher probability of rolling either a 4 or a 5 on 2 dice than rolling simply a 4. Should make intuitive sense. And you should always be doing this kind of mental math to check and make sure that your numbers are not completely made up. So what does this dice have to do with our genetics problems? Well, let's say that you are looking for a situation where you're trying to arrive at children of a certain gender, but you don't care whether they're boys or girls. You just want all of the same gender. Like in my family. My parents, their luck determined that they had 3 boys. So what is the likelihood, what's the probability of having 3 children of the same gender? Now, what are the ways in which you can have 3 children of the same gender? Well, you can have 3 boys, or you can have 3 girls. Both are valid ways in which to have 3 children of the same gender. What's the likelihood of having 3 boys? Well, you need a boy the first child times a boy with the second child times a boy with the third child. This gives you a 1 in 8 chance of getting 3 boys out of 3 children. Now, same with girls. What's the probability of having a girl with your first child? 1/2. Girl with your second child? 1/2. Girl with your third child? 1/2. Giving you a frequency of 1/8 chance of having 3 girls. But because you're not having 6 children, you ultimately need to add these two values together. 1/8 plus 1/8 will result in 1/4 probability, or 25% of the time, you're going to end up with children, or 3 children, of the exact same gender. Do a little bit of a mental check, make sure. Is it 3 children of the same gender? There's only 2 different ways to do that. 25% of the time, I expect the children to be of the same gender, whereas 75% of the time, there should be a little bit of mix and matching. Since there are many different ways to get not 3 boys and not 3 girls, the not situation left for some of your probability classes. Do strongly encourage y'all to take those. We can see, at least logically, that it's less likely for you to have 3 children of the same gender than 3 children of mixed genders. simply because of the number of ways of combining them. So let's take our product and sum rules and the way that we visualize things with our dihybrid crosses and Punnett squares and combine them to something that I guarantee you will not want to examine. What is the likelihood of a genotype given that not all progeny are hybrids, or not all parents are hybrids, and we're looking at 5 loci? So in this situation, we'll have the genotype capital A, lowercase a, lowercase b, as promised, not all hybrids, capital C, lowercase c, capital D, lowercase d, and capital E, lowercase e. Now we're going to cross this against an individual with the genotype capital A, lowercase a, capital B, lowercase b, capital C, lowercase lowercase d, and capital E, lowercase e. So we see that this top individual has 2 lowercase alleles at B, and the bottom individual has 2 lowercase alleles at D. Your task, should you choose to accept it, and hey, you're in this class, so you've accepted it, is to tell me what is the probability of having an individual that is all lowercase at all 5 of these loci. Well, for that, we need to do our Punnett squares again. If I do a Punnett square where I cross these two, the capital A's, lowercase a, capital A, lowercase a, how many of the genotypes are going to be lowercase, our desired genotype? Well, it's going to be 1/4. Let's do the same for all of these. Here, because of the difference in genotype, we see that fully 1/2 of the progeny have our desired lowercase b, lowercase c. 1/4 have it for our lowercase c. Again, 1/2 have it for our lowercase d. And 1/4 have it for our e locus. So we have all of these, and since each has its own independent Punnett square, we'll perform some multiplication. Take a moment, test that on your calculator, make sure your devices work, and you should get a value of 1/256, or represented as a decimal, a very, very small number of 0.0039 trailing. So, I want to illustrate this one example with 5 loci and not hybrids because obviously it's fair game to be dealing with situations where you have varying genotypes. And it is up to your critical thinking to make sure that you are keeping track of all these genotypes and whether they need to be added or multiplied together properly. Now we're going through these lists of different possible ways in which to perform crosses and to calculate the resultant probabilities because ultimately this section is going to be tested in a problem set. Part of genetics is being able to deal with real-world problems and the math associated with them. So you will not get a simple blueprint. You will not be told which way to go. Part of what makes biology challenging as a problem-solving regime, more so than math, or rather in a different way than math, is that it's not always clear which tool you need to be using to hammer in said nail. Not everything is a hammer and not everything is a nail. You have different applications, different situations, and you need to be determining which mathematical path to follow. The math will always be fairly simple. I mean, I'd argue that you can multiply things ever since maybe late elementary school, but it's determining how to derive those values that you're multiplying that makes biology problem solving fun. All right, next up, next class of problems. Well, what happens if you're in a situation where not all of your genotypes survive? What happens if these probabilities aren't always going to be 1/4? Like with lethal genotypes. Do we remember any diseases that happen to have lethal genotypes? Well, yeah, going back to last lecture, from our autosomal dominant inheritance from our pedigrees, you'll remember that pseudoachondroplasia was a homozygous lethal when given the recessive alleles. Now, pseudoachondroplasia was our dwarfism, where lowercase a resulted in a child that would not be born. I will repeat that again. This individual would not be born. So if we're performing our Punnett square, let's say that you're crossing two dwarfs, capital A would end up in an individual whose limbs are not short in any capacity. Lowercase a, lowercase a, capital A. These two individuals would result in dwarf phenotypes. And this individual, lowercase a, would be lethal. That means our expected probability ratios here for children who are born would be 1/3 capital A, and 2/3 heterozygous. For children who are born. You also need to pay attention if instead the question is conceived. Ooh, not that I know how to spell conceived. Is it I before E? I don't know. Let's take a stab. That doesn't look right. That's okay. If we're talking about conception, we have a 1/4 chance of a homozygous capital A, a 1/2 chance of a heterozygote, and a 1/4 chance of our homozygous lethal. In this way, the wording of the question is going to matter a lot. Whether they are born, conceived, or whatever other permutation that they can have on this. Now let's go ahead and combine this with our multi-locus problems, wherein we'll have two trihybrids we'll cross this against itself, indicate it like that, or like this. That B looks quite funky. What is the probability here of conceiving a child or birthing a child with the maximum amount of recessive alleles at each locus? So what is the probability of having a child with a maximum amount of recessive or lowercase, I'll correct that here, should not be saying recessive, maximum amount of lowercase being born? Well, from this Punnett square being born, we see that the maximum amount of lowercase that you can have is 2/3. So 2/3 chance of having the maximum there. Here, you'll have a 1/4 chance of being lowercase b, and a 1/4 chance of being lowercase c. You again can do the math here. When you do the math, you should arrive at a value of 2 over 48, or 1 over 24, or a decimal point. Doesn't particularly matter which. Any problem will state which form it wants to have. Ultimately, it's just entering values into a calculator. So that's another way in which genotypes can be excluded from our possible Punnett squares. Another way that you can combine our lethal genotypes or lethal-like genotypes in a Punnett square is by taking a look at a pedigree. Here you can have a visual of whether an individual is affected or not, and thereby determine their phenotypes if the diseases are recessive. So be aware of that. Lots of different permutations of this, right? Lots of different ways to skin this cat. Now on to a different format of problem, not something that's going to be looking at our ratios within Punnett squares, but rather our odds of sampling, with the meme included, our bad luck Brian. Because binomial sampling wants to look at how many progeny you need to have before a success. It looks at prediction. The number of progeny before having the desired phenotype. Oh, sorry, not phenotype, genotype. Now for this, we're going to have to go back up to one of our crosses. So I'll go ahead and capture this situation and make it a little bit smaller. So if we're looking at this question where you're expecting a ratio of 1 out of every 256, or 0.0039 extended, numbers, a ratio of progeny with the all recessive genotype from, or all lowercase genotype from this cross, we want to figure out how many progeny need to occur, or how many progeny need to be born in order for this all lowercase genotype to be present with some confidence. Well, what is our confidence level? Well, in science, we typically consider a 95% chance of success to be our confidence threshold. You'll oftentimes hear of alpha values of 0.05. This is where you have a 5% chance of failure, so it's the opposite side of a 95% chance of success, as are often quoted standard or metric in terms of statistics. Because this 1 in 256 value is an average of the ratio. It doesn't actually report to you the number of progeny because, like our friend Bad Luck Brian, if you produced only 256 progeny from this cross here, with some bad luck, you might not produce a single individual with all lowercase letters at their genotypes. In order to get an actual expectation of the ratio, or the number of progeny we need to have, we need to look at something called the binomial distribution. The binomial distribution tries to answer the question of how many successes would occur in a number of events where each event asks a yes or no question. In the case of our genetics situation, you're going to be asked for a success rate that differs based on the genetic problem given to you. The binomial distribution is frequently applied to situations of a coin flip, where you can ask how many times do you need to flip a coin. If you flip a coin 10 times, um, what's the probability with that you'll get a certain number of heads, or if you are drawing marbles from a bag and you want to draw one of two different classes of marbles. It's similar to what we're asking with our ratios of an average, but it is instead allowing for sampling stochasticity. Or sampling randomness to play a role here. Now, ultimately, what you're going to see with the binomial distribution is oftentimes called the cumulative distribution function. Whereas the binomial probability mass function does look like your typical bell curve. The cumulative distribution function here, which is oftentimes associated with our binomial sampling, has a situation where it will, approach an asymptote or approach 100% as you get more and more trials. Now, in cases where your frequency or your likelihood of success is something low like 1 in 256, this might be a little bit more of a drawn out curve, but you're still eventually, you have a million children, you're going to get some that have this all lowercase genotype. So how do we actually solve for this? Well, the formula that I'm going to be asking y'all to use is you have to figure out what is the likelihood of not success. So what's the probability of obtaining at least one success is the probability of all possible outcomes minus the probability of failure. This one is all outcomes minus the likelihood of failure, which in our case is 255 out of 256 times you're going to fail. And it's this failure probability that is critical to our binomial sampling. Now, for the rest of the equation, because this isn't yet done, we have to introduce the number of times that we're sampling. The number of times being n. Now, on the other side of the equation, what this must equal is our likelihood of success, or 0.95. That's our 95% confidence interval here. So in this situation, we have 1 minus the so all outcomes minus the probability of a failure set to our success rate given a number of trials. You can see how if you change this, or you set this number of values, let's say that you pull 10 progeny, what is the probability now that you would get 10 progeny that have that all lowercase genotype? So you can solve either for total success rate given a number of draws, or solve for a success, solve for number of draws with a high success rate. So from there, you just need to do a little bit of math in terms of some algebra. You're going to want to take Let's solve this in succession here. You'll want to move this term over to the other side and move over the 0.95 back over to the 1, so we can add 255 over 256 to both sides of the equation. So 1 minus 0.95 is equal to 255 over 256 to the n. You can simplify this down. 0.05 is equal to, how do you get the n out of exponents? Well, you take the natural log. So if we take the natural log of both sides, what happens here? Well, if we take the natural log of 255 over 256, the n, this n is going to pop away as a coefficient to our natural log. You just got to make sure to remember to take the natural log of the left side as well. This will give you a situation where you can have natural log of 0.05 divided by natural log of the probability of failure, 255 over 256, is equal to our number of trials needed to get that success rate. Again, go ahead and plug that into your calculator, and you will see that the number of successes that the number of trials that you need to have is approximately 765 attempts. Now, this is very different than your 256 attempts that you need to expect on average. But this same binomial sampling, and you best believe that you will have some binomial sampling applied in any breeding program or problem set for this class, can be used when changing success rates. You have to make sure that you're identifying the correct number of successes, and you have to identify different thresholds or thresholds of success, or stick with a set number of polls or set number of attempts. I find binomial sampling to be incredibly useful in terms of lots of modeling. So getting comfortable with calculating this binomial sampling and setting up situations is actually something that a lot of population geneticists do in their research. You always want to have some sort of null expectation because our hunches are oftentimes very bad. So instead we set up some math to see if we can improve our hunches and make them a little bit better. You'll get some practice with this during the problem sets. So, a couple other things that will fall into the grab bag of genetics problem solving when dealing with issues surrounding meiotic assortment are the number of unique genotypes. Now, unique or distinct genotypes are something that a lot of scientists care about when performing huge numbers of crosses in terms of strain development or overall calculating diversity. But this isn't asking for individuals of a specific genotype, just the number of genotypes produced, given a set of parents with lots and lots of progeny. So slightly different here. Let's say that we're again given our loci of A, B, C, and D, where all individuals are heterozygous. And you're going to cross them. Now, in this situation, this tetrahybrid cross, the number of genotypes that can be produced at each locus is either homozygous capital A, homozygous capitals, heterozygous, or homozygous lowercase, meaning that there are 3 possible genotypes. The same is true for our B locus, our C locus, and our D locus. Now, since you can have 3 possible genotypes at each of our loci, your final solution is going to be 3 to the 4th. You can go ahead and plug that into a calculator. to derive the number of unique genotypes that can be produced from this cross. Now, this will be slightly different given a different type of cross. This was the value where we had tetrahybrids, but what if instead we wanted to calculate the number of distinct or unique genotypes when performing a test cross? A test cross is where you cross an individual that of an unknown genotype or of a hybrid genotype at each loci against something that is homozygous lowercase at each locus. Now here, you're going to either be able to produce the heterozygous capital A lowercase a or the homozygous lowercase a, meaning that two possible genotypes are created. With two possible genotypes at each location, you will get 2 to the fourth, or 16 distinct genotypes, whereas 3 to the fourth would have yielded you 81 possible distinct genotypes. And so when you're performing these crosses, it certainly benefits you to understand just how many possible genotypes are being produced when looking at different parentage. Something that needs to be in the toolkit of all breeders. Now next up, we're going to do a quick review of chi-squared with a slightly different application, because we'll be looking again at recombination distances and some chi-squared next time, but I want to show you a different way than we've ever applied chi-squared before in terms of looking at probability values. Oh, there we go. Buggy little iPad. So in this situation, or this chi-squared problem, we are again going to be using our great equation, the observed minus expected squared divided by expected to derive at your chi-squared statistic, where we'll have a table with our observed numbers and our expected numbers. And this, all in all, you'll remember, has to be summed across all of your different categories. Now, in this situation, let's go ahead and say we have a bag of marbles to do something different from a genetics problem, so that way you can think independently about how each of these problems are worded, so that way you don't draw a bias from how I'm solving these practice problems and instead approach every single genetics question with your own intuition and make sure that you're setting things up correctly. And we're drawing from a bag of marbles where there are red and green marbles. And you are told that there's 120 of them, and there's 60 of each color. Here, you are being asked to calculate the chi-squared statistic and determine what is the probability, or what percentage of the time would you expect to have the following draws. So slightly different than what you've been asked before. With what probability would you expect the result that you see? To determine that, you're always going to need a chi-squared table. Now, the chi-squared table is going to rely on the degrees of freedom to really pinpoint exactly which bracket you're in. The book, the internet, lots of different places have these chi-squared tables with more or less degrees of freedom and more or less resolution on the probabilities. But ultimately, your degrees of freedom can be understood as the number of classes that can vary. How many independent things are you drawing from? Well, here we're going to have the color of the marble being the thing that varies. We're going to be looking at either red or green. And since we have one class of things that varies, our degree of freedom is going to be 1. Now, in order to solve this, let's go ahead and give ourselves some practice numbers here. You can do the same to your colleagues by changing some of these values. We'll say that I pulled out 55 red marbles and 65 green marbles. I would have expected 60 of each. We can go ahead and throw this into our chi-squared calculator. So you would say 55 minus 65. 55 minus 65 is negative 10. 10 squared is 100. Divide that by our expected number, 60. Except for, actually, wait, I see an error in my math immediately. Our expected number was not 65, it was 60. Therefore, our numerator is not 100, but instead going to be 25. 25 divided by 60 is 0.4167 repeating, or we can go ahead and round that to 0.42. Repeat this for 65 minus 60 squared divided by 60 for our green marbles, and you'll get approximately 0.42. So again, I kind of breezed through that math and made an error in the first place. So make sure that you're able to follow along with this. You can do so and test your abilities by changing these observations. I can give you another problem set where you have, let's say, 40 reds and 80 greens. You can go ahead and solve this afterwards. Now with your math here, you have 0.42 plus 0.42 gives you 0.84 as your chi-squared statistic. Now what does that mean? Well, if we have 1 degree of freedom and our chi-squared value is 0.84, what's our probability of having gotten that number of pulls, that 55 and 65, reds and greens respectively? Well, you can go across this and say, okay, well, 95% chance if our chi-squared value is very, very low, that would be something like 61 and 59. And then keeping on going down, you're going to want to find the two values that 0.84 is in between. Well, 0.84 is between 0.46 and 1.07. Carry that down here. That means it's something between a 50 and 30% probability of having derived these two specific values. We'll split the difference, say something like a 40%, or on a question I would expect you to answer something between a 50 and a 30% chance of having derived those values, those polls. And you can see how if you get an extremely high chi-squared statistic, that it becomes increasingly unlikely that you derive those values from this expected value, and therefore can determine whether or not I'm lying in my expectations. So that's a different way to approach our chi-squared statistics. Again, we'll have a more thorough review of chi-squared in next lecture as it relates to 2-point test crosses and progeny ratios. But for now, we'll be on to our last major topic being discussed in today's assortment lecture, because it's the last one that does not involve recombination. Keep in mind, everything that you've looked at here across the entire lecture has always been dealing with loci on separate chromosomes. Going back to this situation. That's why all of this math is lumped together. It's loci on separate chromosomes. Next time, we'll be looking at recombination. So the last topic here has to do with breeding questions, where we want to breed for homozygosity. Oftentimes in corn breeding, you want to produce inbred lines that have very, very little heterozygosity, so that way you can very easily predict things about their progeny, right? If you have an individual that's totally heterozygous and you cross them against something, you have a huge number of distinct phenotypes and distinct genotypes, right? Ding, ding, ding. We oftentimes in breeding want to self individuals or produce homozygotes from many generations of inbreeding or selfing in order to have predictive genetic ability. Now, this matters a lot because in particular in corn breeding, when we have these inbred lines, we can then take inbred lines and cross them against specific other inbred lines. And when we perform that cross and produce an individual that's entirely heterozygous, we see that phenomenon of heterosis, wherein the child has a phenotype greater than any of the parents and yields much, much more. The phenomenon of 1 + 1 = 3. So, before we get down that breeding tangent, which we'll revisit when we talk about quantitative genetics, what kind of residual heterozygosity remains after you try to self an individual for a large number of generations? So the question here is, if we try to self a heterozygote, we want to ask, per generation, what is the amount of heterozygosity that remains? This allows you to answer the question of, do I need to self this individual one time in order to get most of the heterozygosity gone? Or do I need to self this individual a whole boatload of times in order to get the amount of selfing or heterozygosity to be gone? So the equation for this is fairly simple. The amount of residual heterozygosity is going to be equal to 1/2 times n, where n is equal to the number of generations that you self. To make sure I'm not just speaking in words that you don't understand, selfing is where you take a plant and you cross it against itself. Plants very frequently have both male and female reproductive parts, so this would be taking one plant's pollen and mating it to its eggs and producing offspring. Again, this is different from asexual reproduction or cloning, whereby each progeny is an exact copy of the parent, genetically identical. In this case, you're still performing that independent assortment that will result in, slowly but surely, losses of residual heterozygosity. So, I have a question for you. How many generations would we need to self an individual in order to, a completely heterozygous individual, in order to have less than 0.5% heterozygosity remaining? So at what point does this value drop below 0.005? Take a moment here to solve this. Yep. So you can either do it by solving for n or by trying different values. 1/2 to the 6th, 1/2 to the 7th, 1/2 to the 8th, one half to the 6th gets you a residual heterozygosity of 0.0156%. This value is still 1.5 or 1.6% heterozygosity. Too high. Next up, 0.5 to the 7th gets you below 1% at 0.0078, but still higher than 0.5%. And then 0.5 to the 8th gets you your value of 0.0039 in terms of your genome-wide residual heterozygosity. So that's kind of crazy. You can self a plant for 8 generations. 8 generations of inbreeding with nothing else other than itself. And you're still expected to get a small percentage of heterozygosity. Now we can get into all kinds of corn theory here about how not all parts of the genome are equal in this regard. We see areas where residual heterozygosity is higher than other areas. That's perhaps a discussion for a journal club rather than this lecture. But at least now you understand the math here behind how many generations you need in order to generate that little amount of heterozygosity. Now you'll look particularly intelligent if at some point you interview at a breeding company, a corn breeding company, or any plant breeding company, and they tell you that they perform a number of generations of selfing on each inbred line before they again perform crosses with it with a different parent. You can very intelligently tell them that you know that that number is 8 because it removes all heterozygosity with the exception of less than 0.5%. And less than 0.5% is a pretty tolerable amount. Once you start selfing for 9 generations or 10 generations, you'll notice that the amount of heterozygosity that you remove is less and less. So this is kind of the sweet spot that all corn breeding companies, most of my background is with corn, and it's the plant for which we have the best genetic resources developed, that all of these plant breeding companies, when trying to create what are called inbred lines or highly homozygous lines, they're doing so with historically 8 generations of selfing. Kind of neat, huh? Okay, so that will wrap up our math associated with independent assortment. Please make sure that you try to change up some of the numbers and give yourselves each some practice problems in preparation for our problem sets that we'll be doing together. After all, these lectures are only useful if you can do stuff with that knowledge. So it should be fun. I'll see y'all next time for our discussion of what happens when we throw recombination into the mix.